Hello students, today we will be talking about a topic related to the plant anatomy, and that is the difference in the dicot stem and the monocot stem. This is the backbone of the plant anatomy, and that is the difference between the dicot stem and the monocot stem, right? Very first, I have made a column. On the left hand side you can see I have written the characters, and here I have written the dicot stem, and here I have written the monocot stem. We will be differentiating between the dicot stem and the monocot stem. Very first, what is the epidermis? What type of the epidermis is present in the dicot stem and the monocot stem? See that the epidermis, which is the outermost layer, it is actually having more big size cells, and multicellular hairs are present on the epidermis, and also in the epidermis the stomata which are present are guarded by the guard cells, and that guard cells are kidney shaped. But if you will see here, the cells of the epidermis in the monocot stem are small in size, multicellular hairs are absent, and the stomata is guarded by a guard cell which is having the shape of dumbbell. So you can differentiate it. Cells of the epidermis are big in size, cells are small in size. Here multicellular hairs are present, here the multicellular hairs are absent. Here the stomata is guarded by the guard cells which are kidney shaped. Here the stomatas are guarded by the guard cells which are dumbbell shaped. Now when we see the section of the dicot stem or the monocot stem, just below the epidermis what is present? Hypodermis. Epi means above, hypo means below. The dermis which is present below the epidermis will be called as what? The hypodermis. It's the chief character of the dicot stem that the hypodermis is made up of the collenchymatous tissue, while in the monocot stem the hypodermis is made up of the sclerenchymatous tissue. The third one, that is known as the cortex. Now the cortex is well differentiated here. Basically in the dicot stem the cortex is well differentiated, means it is having endodermis, it will be having the pericycle, pith, pith rays, all are present in the dicot stem. But if you will be viewing the monocot stem, then the cortex is not differentiated here. You will be seeing that endodermis, pericycle, pith, pith rays, all will be absent in the monocot stem. Hence we can say that cortex is not differentiated, but the tissue which is present from the below portion of the hypodermis up to the center is made up of the parenchymatous tissue in the monocot stem, and it is called as what? The ground tissue. So we can say that cortex is well differentiated here, right, and it is made up of parenchyma. Here the cortex is not well differentiated. The tissue which is present from below portion of the hypodermis up to the center of the stem is made up of the parenchyma, and this is called as what? The ground tissue. Now the next part comes known as what? The endodermis. Now endodermis is present here. In the dicot stem the endodermis is present, okay, but the that is the endodermis is not fully developed. It is little bit poorly developed, and it is single layered. The cells are barrel shaped, and Caspin strips are absent. The strips, that is the lignin and the suberin strips known as the Caspin strips, are absent here. Only the starch grains are present in the endodermis of the dicot stem, and that's why we can also call it as that endodermis here is also called as the starch sheet. But in the monocot stem the endodermis is altogether absent. So this is also a differentiating feature, that endodermis is present here, though it is poorly developed. It is single layered, cells are barrel shaped, Caspin strips are absent, but starch grains are present. Because of the presence of the starch grains, the endodermis can also be called it as starch sheet, okay, but in monocot stem the endodermis is altogether absent. Now the next comes the pericycle. Pericycle is basically present in between the vascular bundles and the endodermis, okay, and we can say that this pericycle is boundary layer. Pericycle is boundary layer of the stele. It is the boundary layer of the stele, and it is present in the dicot stem. But what is the feature of this pericycle? the pericycle is present in the dicot stem and it is multilayered and it is made up of parenchyma and sclerenchyma, means it is heterogeneous, means pericycle is heterogeneous in nature in the dicot stem and it is made up of parenchyma and sclerenchyma. But in monocot stem the pericycle is altogether absent. So these are the differentiating features. Some more features are there, you just have a look here. Now coming to the sixth point, and we'll be discussing about the stele, that what type of the stele is present in dicot and monocot stem. So this is the sixth point, and that is known as the stele. Okay, stele. Now all the portion which is present inside the pericycle will be called as what? The stele. I am making the stele here which is found in the dicot stems. Okay, now this is a stele. I made the stele here, and you can see that here the vascular bundles are present in a ring. Here the vascular bundles are present in a ring, and such a stele which is found in the dicot stem will be called as U stele. What is U stele? Vascular bundles are arranged in ring. They are arranged in ring. Okay, and in the case of the monocot stem, the type of the stele is atecto stele. Atecto stele means what? That here the vascular bundles are scattered. They are not present in a ring. They are scattered here and there. Okay, here the vascular bundles are scattered here and there, and such a stele is called as atecto stele. So what is the difference between the stele in the dicot and the monocot stem? In dicot stem the stele is U stele type, in which the vascular bundles are present in a ring, while in the case of the monocot stem the stele type is atecto stele, means vascular bundles are scattered. Okay, next point. Now coming to the next point, and that is known as the vascular bundles. That what type of vascular bundles are present? What type of vascular bundles are present in the dicot stem and the monocot stem? So you will see that vascular bundles are conjoint, collateral, or sometimes they are bicollateral, endark, and open. For more details that what is conjoint, collateral, bicollateral, endark and open vascular bundles, you can see my video that is by the name types of the vascular bundle, in which you will be able to know that what is conjoint, collateral, bicollateral, endark and open vascular bundles. Okay, so vascular bundles means conjoint, collateral, bicollateral, endark or open. While here in the case of the monocot stem, the vascular bundles are conjoint, collateral, endark and closed. Why closed? Because here the cambium is absent, while here the vascular bundles are open. Why? Because the cambium is present. So the type of the vascular bundle, what is the type of the vascular bundle? Conjoint, collateral, sometime bicollateral, endark and open. While in the monocot stem the vascular bundles are conjoint, collateral, endark and closed. You can also learn by the code language CSIR, means conjoint, collateral, endark and closed. Here I am writing the code words CCEO, means conjoint, collateral, endark and open. While in some cases, such as in the cucurbita stem, the condition will be conjoint, bicollateral, endark and open. Conjoint, bicollateral, endark and open. And in maximum stems, in the maximum dicot stem, the condition is conjoint, collateral, endark and open. While in the monocot stem, the vascular bundles are conjoint, collateral, endark and closed. Okay. Now, what is the shape? What is the shape of the vascular bundles? What is the shape of vascular bundles? So here the shape of the vascular bundles is wedge shaped. The shape is wedge shaped, while here in the case of the monocot stem, the shape of vascular bundles, the shape of vascular bundle is oval. This is the difference. Here the shape is wedge shaped and here the shape is oval. What is wedge shaped? Wedge shaped is just like a nail, like this type. Okay, while here the shape of the vascular bundle is oval. The shape of the vascular bundle is oval. Okay, what about the size of vascular bundle? Size, all the vascular bundles are of same size. All vascular bundles are of same size. In dicot stem, all the vascular bundles are of same size, but in the case of the monocot stem, vascular bundles present at periphery are small in size, while those present at center are big in size. You can see in the steel region I have made the vascular bundles, and at the center part you can see that I have made big vascular bundles, while at the periphery I have made small vascular bundles, which suggest and say that the vascular bundles are not of the same size. The vascular bundles present at the center are big in size, while those present at the periphery are small in size, while here all the vascular bundles are of same size. Okay, and all the vascular bundles are present in ring that we have discussed here also. That's why we call it as U steel. And here the vascular bundles are scattered. Here the vascular bundles are scattered. Okay, so vascular bundles are conjoint, collateral, bicollateral, and dark and open. Here conjoint, collateral, and dark and closed. Shape is wedge shape, shape is oval. Size, all vascular bundle of same size, they are of unequal size. They are present in the ring, they are scattered. Okay, now coming to the next one, that what is the bundle sheet? What is the bundle sheet? So coming to the next point, 8th point, that is bundle sheet. Bundle sheet. You must know that bundle sheet is present around vascular bundle, but it's not necessary that bundle sheet is present in all the vascular bundles. Here in the case of the dicot stem, the vascular bundles are without bundle sheet, means bundle sheet is absent. Vascular bundles are not surrounded by bundle sheet, but here bundle sheet present, and this bundle sheet is made of sclerenchyma, means the vascular bundles are surrounded by bundle sheet which is made up of the sclerenchyma, but in dicot stem the bundle sheet is absent. Now coming to the next point, that xylem. Now xylem consists of more xylem vessels. Xylem consists of more xylem vessels. Here xylem consists of less xylem vessels. Okay, and they are present in V or Y shape, while they are present in radial fashion. They are present in radial fashion. Okay, so xylem, here the xylem consists of more xylem vessels and they are present in radial fashion. Here the xylem consists of less xylem vessels and they are present in V or Y fashion or V or Y shaped condition. Okay, 10th one, phloem. As we know very well, xylem and phloem are found in the vascular bundles. That's why we are discussing phloem. So in the case of the dicot stem, phloem consists of phloem parenchyma. So we can say that phloem parenchyma is present, while here the phloem is devoid of phloem parenchyma, means altogether phloem parenchyma is absent. There is no monocot which consists of what? The phloem parenchyma. So here the phloem parenchyma is present and here the phloem parenchyma is absent. Okay, next comes cambium. As you know very well that there are some vascular bundles in which in between the xylem and the phloem cambium is present, and when cambium is present the vascular bundles are called as open, and when the cambium is absent the vascular bundles are called as closed, right? And cambium is responsible for the secondary growth. Okay, so in dicot stem cambium is present. Say for I have made a vascular bundle, right, and here I have written phloem and here I have written xylem, and in between this xylem and the phloem what is present? This is the cambium. Okay, this is the cambium. So here the cambium is present. That's why the vascular bundles are open type. While here in the case of the monocot stem, cambium is absent. Altogether it is absent. Means say for if this is the vascular bundle, then in between the phloem and the xylem cambium is not there. So cambium is absent there, and in the case of the dicot stem cambium is present. Okay, now coming to the next one, as we know very well cambium is responsible for the secondary growth. So let's have a look that secondary growth will be present in the monocot or the dicot. So secondary growth. Now as we know very well here cambium is present, and that's why what happens, secondary growth occur in the case of what? The dicots, while here secondary growth is absent. This is the main feature of the monocot stem, that in them secondary growth is absent. Why secondary growth is absent? Because vascular bundles are devoid of cambium, and cambium is responsible for secondary growth. Now if cambium is absent, so definitely the secondary growth will also be absent. Okay, so this was about the secondary growth. Now coming to the next few points also between the dicot stem and the monocot stem. Now point number 13th. See, now coming to the next one, that is pith, that whether the pith is present in the dicot stem or not. So here well defined means developed, well developed pith present. It is made of parenchyma with lots of intercellular spaces, and it is meant for storage. But here, as we know very well that the cortex is not well defined, it is not well differentiated, and hence we can say that pith is absent here. Pith is altogether absent. No pith is present in the case of the monocot stem. The next point, pith rays. Pith rays means what? Medullary rays. These pith rays are present between the vascular bundles. Pith rays are present between the vascular bundles. So as the pith is present here, so here also the pith rays in dicot stem, pith rays are present. Pith rays present and are meant for lateral conduction of food and water. Okay, but here in monocot stem, as the pith is absent, so here pith rays are absent. In between the vascular bundles there are no pith rays. Okay, and the next point, lysogenous cavity. Lysogenous cavity is basically here water cavity. If you will see the vascular bundles, so just below the vascular bundle, in the case of the monocot stem, particularly in the case of the monocot stem, you will be able to see a water cavity just below the vascular bundle, but in the case of the dicot stem, you will not be able to see the water cavity. So we can say that here water cavity, which is a type of a lysogenous cavity, water cavity is absent in dicot stems, while here in the case of the monocot stem, just below the vascular bundles, water cavity is present. Okay, so these were the 15 differences between the dicot stem and the monocot stem. What you have seen here, that in the case of the monocot stem, endodermis, pericycle, pith and pith rays are totally absent. This must be remembered. While in the case of the dicot stem, the endodermis, pericycle, pith and pith rays all are present. Right? We'll be coming soon with few more videos on plant anatomy, which will be based on the differences between the dicot root and the monocot root, and the differences between the dicot leaf and the monocot leaf. So keep watching my videos, subscribe my channel, also like and share and comment. Thank you.